Good day everyone. We're the third group and we will be discussing our report entitled The Genetically Modified Organisms. Before we begin the discussion, please allow me to introduce myself and my team members. I'm Angeline Imparagas and I'll be discussing about the significance of GMO, biotechnology, and applications of biotechnology. First, what is GMO? A genetically modified organism, or GMO, is an organism whose genetic material has been altered using genetic engineering techniques, a source of genetically modified foods, and is also widely used in scientific research and to produce useful goods other than food. Organisms that have been genetically modified include microorganisms, such as bacteria, plants, fish, and mammals. Biotechnology is the manipulation of biological organisms to make products that benefit human beings. It contributes to such diverse areas as food production, waste disposal, mining, and medicine. Modern achievements of biotechnology include the transfer of a specific gene from one organism to another by means of transgenics. Transgenic plants. In research, plants are engineered to help discover the functions of certain genes. One way to do this is to knock out the gene of interest and see what phenotype develops. Another strategy is to attach the gene to a strong promoter and see what happens when it is overexpressed. In transgenics, the maintenance and growth of genetically uniform plant and animal cell cultures called clones, and the diffusing of different types of cells to produce beneficial medical products such as the monoclonal antibiotics. The first achievements in biotechnology were in food production, occurring about 5000 BC. Diverse strains of plants or animals were hybridized or crossed to produce greater genetic variety. Repeated cycles of selective breeding produce many present-day of food production programs. In the 1960s, an important project used hybridization followed by selective breeding to increase food production of quality of wheat and rice crops. Applications of biotechnology. Biotechnology has been applied in various fields, in medicinal, agricultural, and food. Medicinal biotechnology. In medicine, biotechnology has been extensively applied in the development of many drugs, gene therapy, gene testing, immune system, and many other treatments. Biotechnology is also used to develop various vaccines, such as insulin. Agricultural biotechnology. In this technology, scientists develop new crops by incorporating genes that code for important characteristics. Scientists also splice the superior gene from the one plant and incorporate it into another. GMO crops have been highly successful in Africa and the Middle East. In food biotechnology, biotechnology is involved with the infusion of nutrients into food products, and it is also involved with modification of enzymes. These enzymes are used in making bread, syrups, cheese, meat tenderizing, and etc.
Hello, my name is Giancarlo Sarmiento and I will explain to you what is recombinant DNA technology and its techniques and uses of data profiling. So let's go. Genetic engineering or recombinant DNA technology and its technique. Recombinant DNA technology. Genetic engineering is the alternation of an organism genetic or hereditary material to eliminate undesirable characteristics or to reproduce desirable new ones. Genetic technology. Knowledge from genetic studies applied to medical research, criminal investigation, agriculture, and other areas. Selective breeding. The first agricultural technique is still utilized. Enhance food production by choosing plants and animals with desired traits for breeding. Now we're done about genetic engineering or recombinant DNA technology and its technique. DNA profiling or fingerprint. This is also termed as DNA profiling, wherein the process itself produces unique genetic map or fingerprint for each individual. Each enzyme splits DNA at different sites, resulting in different fragments of different lengths. These fragments form a pattern resembling supermarket barcode, which can be matched with another DNA fingerprint sample. Let's proceed to uses of DNA profiling. Number 1. Hospital genetic testing diagnoses inherited disorders like cystic fibrosis and sickle cell anemia in newborns and prenatal cases worldwide. Number 2. It helps predict prospective parents understand the risk of having an affected child. Number 3. By the DNA fingerprints or relatives who have a history of some disorders, it is possible to identify DNA patterns associated with the disease in question. Number 4. DNA fingerprints link suspects to biological evidence. Blood or semen stains, hair, or clothing found at the scene of a crime. Number 5. DNA fingerprints establish paternity in custody and child support litigation. At last, number 6. DNA fingerprints help identify casualties or persons missing in action.
Hello everyone, my name is John Rafael Javier. I'll be covering my topic, gen splicing, application of gen splicing, food production, population control, medicine. Gen splicing. Scientists used to directly alter genetic material to form recombinant DNA in gen splicing. One or more genes of an organism are introduced to a second organism. In gen splicing, DNA cannot be transferred directly from into original organism known as the donor. The genetically engineered products in humans, other animals, plants, bacteria, or viruses. Example, gen splicing is a post-transcriptional modification in which a single gene can code for multiple proteins. Application of gen splicing. Food production. Recombinant DNA is used to combat one of the greatest problems in food production, the destruction of crops by plant viruses or insect pests. Example of food production. The problem in food production is the plant viruses or insects, pests, but also scientists develop transgenic potato and strawberry plants that are frost resistant. Population control. Genetically altered bacteria can be used to decompose garbage and to break down petroleum products. Example of population control. The bacteria of garbage can be used to decompose many forms, and also the recombinant DNA can be used to monitor the breakdown of populants. Medicine. It is an important genetically engineered drug is interferon, a chemical produced by the body in a tiny amount. Scientists also produce medically useful human proteins in animal milks. Example of medicine. It's very important genetically any drugs is interferon. Also scientists produce medically useful for human proteins in animals' milks. Sheep milk is used to produce alpha antitrypsin. Cow milk is used to produce lactoferrin. Goat milk is used to produce TPA, a blood that dissolving.
My name is Gerard Bernes. I will be covering classification of gene splicing. Applications of gene splicing. Cloning. Scientists remove the DNA containing nucleus from a female's egg and replace it with a nucleus from an animal of a smaller species. Applications of gene splicing. Why we do cloning? To ensure the replication of desirable traits consistently across generations. It helps maintain specific genetic characteristics such as resistance to pests or disease, increase yield, or improve nutrition content in crops or organisms. Applications of gene splicing. Genetically engineered organism. Unmodified organism through genetic engineering. Combining DNA molecules from various sources to create new genes and transfer them into the organism. Transgenic organism. It is a subject of GMOs of organisms which have inserted DNA that originated in a different species.
Hi everyone, my name is David Sarvaras. I will cover the necessity of GMOs in this presentation. Let's dive in. GMOs are widely utilized in medical research, pharmaceutical drug production, experimental medicine, and agriculture.
Ang transgenic microbes sa genetic-modified bacteria ay mga mikrobyo na binago ang kanilang gene makeup sa pamamagitan ng pag-introduce ng mga gene mula sa ibang organismo. Ang transgenic microbes at genetic-modified bacteria ay ginagamit sa mga pag-aaral ng mga biological process at pag-uunawa sa mga genetic mechanisms. Maaaring gamitin sa pagdevelop ng mga uri ng mga gamot at bakuna. Ang mga ito ay maaaring magdulot ng mga katangian tulad ng resistensya sa mga sakit o pagtaas ng epektibidad ng mga gamot. Ang transgenic animals ay mga hayop na mayroong genetically modified na DNA. Ang mga ito ay nilikha sa pamamagitan ng pag-insert ng mga gene mula sa ibang species sa DNA ng hayop upang magkaroon ito ng mga bagong katangian o mga pagbabago sa kanilang genetic makeup. Ang proseso sa paglikha ng transgenic animals ay karaniwang ginagawa sa pamamagitan ng teknikang tinatawag na gene engineering. Sa pamamagitan ng paggamit ng mga teknikang tulad ng DNA sequencing, gene splicing at gene transfer, maaring maipasok ang mga gene mula sa ibang species sa DNA ng target na hayop. Ang transgenic fruit flies o mga bungang langaw na binago ang kanilang genetic makeup ay mga organismo na ginagamit sa mga pag-aaral ng mga biological process at pag-unawa sa mga genetic mechanisms. Ang mga ito ay mga gene na idinadagdag, binago o tinanggal upang maunawaan ang mga proseso ng paglaki, pag-unlad at pag-iba pang mga katangian ng mga langaw. Transgenic fish o mga isdang binago ang kanilang genetic makeup. Ang mga transgenic fish ay maaaring magkaroon ng mga katangian tulad ng mas mabilis na paglaki, mas mataas na pagdami, o mas matibay na resistensya sa mga sakit. Sa pamamagitan ng pag-introduce ng mga gene, nagbibigay ng mga katangiang ito. Maaaring mapabuti ang produksyon ng mga isda sa mga pangisdaan. Ito ay maaaring magresulta sa mas malaking kita para sa mga mangingisda o mga negosyante sa industriya ng isda.
Hi everyone, my name is Sandara Subang. I'll be covering the issue and concern of GMO in this presentation.
How can we determine if our food is GMO by looking the label or packaging? By looking the label like non-GMO, Project Verified, or United States Department of Agriculture, organic and reduced ingredient list for GMOs like soybean and corn, as some countries mandate GMO labeling. Ang Non-GMO Project ay isang non-profit na organisasyon na nakatuon sa pagbuo at pagreserba ng non-GMO na supply ng pagkain para sa lahat. If GMOs are safe to be consumed, GMO crops and fishes are safe for human consumption due to thorough testing and lack of adverse effect as agreed upon by the National Academy for Science and Health Organization. Ang mga pananim at isda ay ligtas dahil sa pagkonsumo sa GMO nito sa pamamagitan ng pagsubok at napatunayang walang masamang epekto. If they pose environmental threats by introducing them in underdeveloped countries. Genetically modify crops by reduced biodiversity in regions with high genetic variation, potentially causing irremediable gain escape through pollen and raise concerns about pests and weeds resistance. Maaaring bawasan ng mga genetically modified crops ang biodiversity sa mga relihiyon na may mataas na genetic variation na posibleng magdulot ng hindi nalulunas na genic escaper. If biotechnology corporations are efficient in controlling most of the technology needed to develop GMOs. Genetically modified organisms can migrate and proliferate globally, requiring clear signals and biotechnology laboratories to avoid unattended harm to public health, biodiversity, and the environment. Nangailangan ng malinaw na senyales sa mga laboratorio ng biotechnology upang maiwasan ang hindi sinasadyang pinsala sa kalusugan.